Greetings class, Menero Online here. We are carrying on with the monohybrid crosses. So today we're going to cross two heterozygous red flowers. So we always start with P1 phenotype. P1 meaning parent number one. So here we will say heterozygous red. We cross it with another heterozygous red flower. Let's make it a small letter here. Okay, so from there, the next step, we are going to genotype. Remember, if the red color is dominant, we are going to have two options for red color for genotype, which can be two capital letter R's or a capital letter and a small letter. So since we are having heterozygous, it's going to be a capital letter and a small letter. So this is genotype. This is our other genotype. The next step that follows is going to be our meiosis. It's very important for you to indicate it. It's going to be meiosis. Then meiosis will be followed by the gametes. Meiosis will be followed by the gametes. The meiosis separates the gametes. Then we have capital letter R here. We have small letter r. Multiplication to indicate that they are crossing. Then we separate this one, capital letter R. Then this one also is get separated. Then it's going to be the small letter r. Carrying on, the next step here. Remember, before we go to the next step, we must label this number 1, number 2, number 3, and number 4. That will remind you whether you have crossed everything or not. So the next step that follows is fusion. It's fusion. Another name for fusion is fertilization. So after fusion, we are going to have F1 genotype. F1 genotype. So with this one, we will cross number 1. It's going to cross with number 3. I'll write it in brackets here. Inside it's 1 and 3. So number 1 is capital letter R. Number 3 is also capital letter R. Next, we go here, number 1. We are crossing it with number 4. Number 1 and number 4. We are crossing them together. So it's capital letter R and small letter r. We go to the third one. We are done with number 1. Now it's going to be number 2, crossing with all the letters on the other side. So we will start here. Let's use a green one. We'll start here, capital small letter R and capital letter R. Some learners here, they made a mistake and make a mistake and start by putting small letter. That is not the case. We always start with capital letter. So it's going to be capital letter R and small letter r. So here we were crossing number 2 and number 3. Number 2 and number 3. I think next time I should write the numbers down here so that you can see them properly. So the last one, it's still this one, number 2 again. It's crossing with number 4. Then we have small letter r, small letter r. Let me just indicate it here. This is between number 3 and number 4. I think you can see it easier here. This is 2 and 3. Then this one is 1 and 4. And then this one is 1 and 3. I think they are clearer here. So the last step that we are going to, we are still on F1, but this time we are doing the phenotype. Remember, phenotype we use words, genotype we use letters. So if it's capital letter R is going to be red. Capital letter R, small letter r, it's heterozygous red. Capital letter R, small letter r, is also going to be a red as well. Then the last one, we have 2 small letter r's. Then this one is going to be white because it's recessive.